Hello friends, how are you doing? And I hope you all are doing good. And I think you revised we did last time. We are into the chapter. Which chapter we are in? We are doing transport in plants. Right? We are doing this chapter, transport in plants. And now we talked about the reasons for the ascent of sap. The first one we told was the root pressure. We said that the root pressure is due to what? Due to enough water being because we said the root pressure is due to continuous absorption of water by the root hairs, and there is no loss of water by transpiration from the aerial parts of the plant. As a result of this, what happens? A continuous column of water gets on absorbing, and this creates a pressure called the root pressure. This root pressure, however, was able to or in fact is able to conduct water only up to a few centimeters, and hence is useful only for small herbaceous plants. Okay, I'm sorry. Transpiration. Pull is the another factor which we can talk about. Transpiration pull. Now, what is transpiration? Transpiration, first of all, transpiration is a loss of water from the aerial parts of the plant from the stomata. Transpiration is loss of water, right? Transpiration is loss of water in the form of water vapor, in the form of water vapor from the aerial parts of the plant, from the aerial parts of plant. Okay. So transpiration is loss of water in the form of water vapor from the aerial parts of the plant. And transpiration takes place from the stomata. Transpiration takes place from the stomata, right? So transpiration takes place from the stomata. And next thing also, transpiration is the price which the plant has to pay for photosynthesis. Transpiration is the price which the plant has to pay for photosynthesis. Okay. So transpiration is the price which the plant has to pay for photosynthesis, and transpiration is loss of water from the aerial parts of the plant. Transpiration takes place from the stomata, and transpiration is the price which the plant has to pay for photosynthesis. So how does it happen? It happens from the stomata. And where is the stomata? That is the opening present on the leaves. So that stomata actually is guarded by the guard cells. We'll see how. So how does this stomata open? So what happens? See, we are little understanding things which we could have understood in photosynthesis chapter, but anyways this will do. This is there. So the opening and closing of stomata, there in your syllabus, the opening and closing of stomata is guarded by guard cells. Okay, the stomata are opening and this is guarded by the guard cells, and these guard cells are kidney shaped. These guard cells are kidney shaped in dicots, and dumbbell shaped in monocots. Okay. So the opening and closing of stomata is guarded by the guard cells. The guard cells are kidney shaped in dicots and dumbbell shaped in monocots. So what we want to understand, what do we want to understand over here? So when, say, I'm drawing the guard cell, right? If I'm drawing the guard cell, okay, now what we see here is, these are the cellulosic microfibrils. Okay, this is the chloroplast. This is what? The subsidiary cells. This is the stoma opening. All right, the inner wall is thick. And which is the guard cell actually? This is the guard cell. Okay, these two are the guard cells. These are the epidermal cells called as epidermal cells, called as subsidiary cells. This is one. Okay, these are the chloroplasts I have mentioned. All these are chloroplasts. This is a stoma opening. The inner cells are the inner wall is thick when we talk about the dicot guard cells. Dicot plant. Can you see this till here? So guard cells in dicot plant. Guard cells in, right? The guard cells in the dicot plant. So what we see here, as I told you, the guard cells in the dicot plant will be bean shaped or kidney shaped, right? The inner wall is thick and the outer wall is thin, and these inner and outer wall are they are guarding the stoma opening. So what happens when the guard cell has to open? When the guard cell has to open, the this opens only, or we can say the stoma opening, this opens only when the guard cells are turgid. And how does the guard cells become turgid? See, there are two concepts or two hypotheses related to this. One says that the guard cells have chloroplast, and these chloroplasts are absent in these epidermal cells or subsidiary cells around these guard cells. Okay, so this hypothesis, this one particularly says that these chloroplasts at the guard cell manufacture, they perform photosynthesis because of the chloroplast. These guard cells perform photosynthesis, manufacture glucose, and as a result the tonicity of these the guard cells increases. And since now the guard cells are more hypertonic as compared to the other subsidiary cells, so what will happen? Water will move from epidermal, that is subsidiary cells, to the guard cell, and it will become turgid. And when it becomes turgid, what happens? The outer and the inner wall open, and the stoma opening is exposed. So when is this now the stoma opening getting exposed or open? During the morning, when photosynthesis is happening, because of the chloroplast contained in the guard cell. So we can say, I'll write it here, the guard cells open, or we can say manufacture, the guard cells manufacture food with help of chloroplast. Okay, because they will be having the chlorophyll pigment where in the thylakoid membrane. So the guard cells manufacture food with the help of chloroplast. This food, which is nothing but glucose, will increase the osmolarity, will increase the hypertonicity, of guard cells as compared to epidermal cells, hence endosmosis in guard cells, So guard cells become turgid, and open, and the stomata opens. The other hypothesis, so what is this? The guard cells manufacture food with the help of chloroplast. This happens during the morning because this is photosynthesis happening. This food, that is glucose, will increase the hypertonicity of the guard cell. Okay, this is important, will increase the tonicity as compared to the epidermal cells, hence endosmosis in guard cells. So the guard cells become turgid and the stomata opens. The next hypothesis says that these epidermal cells, the next hypothesis says that these epidermal cells, called the subsidiary cells, will actively take in sodium and potassium ions, and then what will happen? Then there will be exosmosis from the guard cell, and hence the stomata will close. Right. The closing of stomata. Right. define wall pressure. These are all 1 mark question. So, we will see what is wall pressure. Okay, wall pressure. Now, one thing we should know that both the wall pressure and the turgor pressure exerted are equal and opposite, equal in in magnitude, but opposite in direction, right. So, what we say that when a cell gets turgid, okay, when a cell gets turgid, right, the cell contents will exert pressure on the wall, will exert pressure, okay, will exert pressure on the wall, and this is called as the turgor pressure, and this is called as turgor, okay, and this is called as turgor pressure, right. Why I talked about turgor pressure? Because now I will be able to explain you better the wall pressure. However, the pressure exerted, right, however the pressure exerted by the wall back on the cell contents is called as the wall pressure. Back on the cell contents is called as wall pressure. So, when a cell gets turgid, the cell contents will exert pressure on the wall, and this is called as the turgor pressure, the pressure exerted by the wall back on the cell contents. So, you see they are actually what if this is a cell and we place it in water, a pure water solution, right. This is pure water, there will be endosmosis, right, and because of this endosmosis what will happen, this will get turgid, the cell will get turgid. So, the cell contents will exert pressure on the wall, this is called the turgor pressure, TP, what? TP from the nasal tang, okay, and the wall will exert pressure back on the cell content called as the WP, wall pressure, yes, the wall pressure, okay. There should be some amount of fun going on. Next, name the pores through which guttation occurs. Beautiful. The pores through which guttation is what? The loss of water in the form of water droplets from the aerial parts of the plant, and this process of guttation happens whenever there is excess of root pressure. Takes place in small herbaceous plants, especially in the morning. What happens due to excess amount of water? At the night, what happens? There is excess amount of water absorption, there is no loss of water, the amount of water inside the plant increases, and that excess water is lost from the pores by the process of guttation, and this it takes place through the hydathodes. The guttation takes place through pores called hydrathodes. The guttation takes place through pores called the hydrathodes, and name the pores through which the guttation occur. Okay, we'll move on to the next question. We'll move on to the next question. What is transmembrane pathway? The word itself is telling you transmembrane, transmission across the membrane. So any movement, any transport, any movement across the membrane will be called as the transmembrane pathway. Okay, any movement across the membrane, right, is called as transmembrane pathway. Right? Any movement across the membrane is called as a transmembrane pathway, and this is what we know. Right. Define water potential. Water potential. Water potential may be defined as a potential of pure water, or any solution for that matter. Or we can say that water potential is the energy possessed by the water molecules. Okay, so this is the potential or energy. This is the potential or energy possessed by the water molecules. Okay, so this is the potential or energy possessed by the water molecules. Now what happens if the water potential of pure water, okay, the water potential of pure water is 0, of pure water is 0. When you add any solute to water, when you add any substance to water, the water potential decreases. On addition of any solute, the water potential decreases below 0. On addition of any solute, the water potential decreases below 0. Why? Because now, because now some of the water molecules have been engaged by this solute, so the energy, the task which the water molecule can perform has reduced, and that is why we say that the water potential of pure water is 0, and on addition of any solute the water potential decreases below 0, that is it becomes negative, that is it will become negative, right. We move on to the next question. Which fraction of soil water are readily available to the plants for absorption? Yes, it is the capillary water which is readily available to the plant for absorption. the capillary water is readily available, right, to the plants for absorption. Right, the capillary water is readily available to the plants for the absorption, and this is what we say, which fraction of soil water, fraction of soil water are readily available to the plants for absorption. Right. Which mention two ways of absorption of water in plants. Good. The two ways, okay, we are doing the sixth question, of absorption of water in plants. The two ways of absorption of water in plants are, one is the apoplastic pathway, and the other one is the symplastic pathway. Right, the apoplastic pathway and the symplastic pathway, and what will we see in the apoplastic pathway? Now, in the apoplastic pathway, what we see, that there is a movement of water through the cell wall and the intercellular spaces. Movement of water right through cell wall and intercellular spaces, right, and intercellular spaces. However, in the symplastic pathway, we will see that there is movement of water within, okay, within the cell, and then from one cytoplasm of the cell to the other by channels called as the plasmodesmata. So, there is transport of water, there is movement of water from one cytoplasm of cell to another through plasmodesmata channels. Plasmodesmata channels. Okay, so these are the two ways of absorption of water in plants. We move on to the next question. What are the factors affecting the rate of diffusion? Right. What are the factors affecting the rate of diffusion? Factors affecting the rate of diffusion. Factors affecting the rate of diffusion, the first of them is temperature. The first of them is the temperature. The more is the temperature, the more energy the diffusing particles will have, because the kinetic energy has increased and hence the rate of diffusion would be higher. Okay? The more the temperature, the more the temperature, the more kinetic energy, the particles will have, and hence higher rate of diffusion. Okay, and hence higher rate of diffusion. Secondly, factors affecting the rate of diffusion, first of all is the temperature. Secondly, we will have the what? The density of the diffusing substance. Right? Density of the substance in which the diffusion is happening. So, density of the medium or substance in which diffusion is happening, right. The lower the density of the substance in which the diffusion is happening, the more will be the rate of diffusion. And fourth, the diffusion pressure, the diffusion deficit. Okay, the what is the concentration deficit? Concentration deficit. What do you mean by this? Say, you know, this is a medium to which there are 80 particles here, say, and there is just 10 particles. The difference is 70. Okay, concentration deficit, that's what. So, and say this is another example B, and this is say A, and say there is 80 particles here, but here it is 60. So, the diffusion pressure deficit is 20. So, what will we see? The rate at which diffusion is happening in A is much more than as compared to in B. Yes, but here the deficit is more, and hence the diffusion rate will be more. Yes, so these are the factors affecting the rate of diffusion.